Multiple sequence alignment is almost the most helpful tool in bioinformatics. In this video, I will talk about the meaning of multiple sequence alignment, or as it is abbreviated MSA, and why it is important in bioinformatics. So the outline for our lecture: first we will talk about the definition of multiple sequence alignment, and then we'll talk about the purpose or the importance of this method. And by the end of this lecture, you will be familiar with the Clustal software, and hopefully you will be able to perform multiple sequence alignment using it. Then from the results of multiple sequence alignment, you will construct a phylogenetic tree, and we will analyze it together. By definition, multiple sequence alignment is generally the alignment of 3 or more biological sequences. This alignment will achieve the optimal matching of the sequences. Look here at the figure. You can see that aligned sequences are typically represented as rows, and gaps are inserted between the residues so that identical or similar characters are aligned in successive columns. We can say that sequence alignment is the process of comparing and detecting similarities between biological sequences, and these regions of similarities may be a consequence of functional, structural, evolutionary relationships between sequences. Now let's talk about the applications of multiple sequence alignment. First, it is used to reveal functional residues and motifs. Also, we use it to enhance the accuracy of structure prediction. Multiple sequence alignment can give you the almost perfect protein or RNA secondary structure. Sometimes it helps even with the three-dimensional structure. Another important application is phylogenetics. Phylogenetics is the study of evolutionary history and relationships among or within groups of organisms. It is mainly concerned with the relationships of an organism to other organisms according to evolutionary similarities and differences. And multiple sequence alignment is the first step and the most critical in building the phylogenetic tree. The result of multiple sequence alignment is the precursor to construct this tree. Another interesting application of multiple sequence alignment is designing PCR primers. So usually in PCR we have the sequence that we want to amplify, and we design two primers, the forward and reverse, for it. But another creative method that allows you to use two primers for more than one sequence is multiple sequence alignment. For example, if you have a sample of unknown bacteria, now if you don't know the identity of the bacteria, you can't design primers for it. But one thing we can do is to use the universal region in all bacteria, which is the 16S ribosomal RNA. When we align the 16S ribosomal RNA for the known types of bacteria, after the alignment of the sequences, we can find a region that can be used for primers forward and reverse, and these primers can bind to most types of bacteria. Now this primer is called universal primer, and the designing of universal primer needs multiple sequence alignment. Finally, we can use it to classify proteins into families. Multiple sequence alignment can help you to decide that your protein is a member of a non-protein family or not. Now let's take an example. We will perform multiple sequence alignment for the primary amino acid sequence of cytochrome C from the following species: Mus musculus, Saccharomyces cerevisiae, Arabidopsis thaliana, Drosophila melanogaster, and Homo sapiens. So first we are going to use a website called NCBI to get the sequence of the protein cytochrome C. This is NCBI. Make sure that you are in protein, and here write the name of the protein with the species. When you go down there, you'll find the sequence of the protein. I'll take it, I'll copy it in my Word file, and do the same thing for the other species. Now we have the Saccharomyces, do the same thing, take the sequence, copy it, and paste it here. The third species is Arabidopsis thaliana. Copy the sequence, and paste it. Melanogaster now. Drosophila melanogaster. Drosophila. And finally, the Homo sapiens. Now, after we get our sequences, we will go to the multiple alignment program, which is Clustal Omega. Make sure that you are in protein, because we are working on amino acids. And now in this box, I will copy the sequences. I will copy them and paste them here in the box. Now we are ready for submitting. Here's the result. Here we have the 5 species, And what we have in the top is the amino acid sequence here. This is the amino acid sequence, and here this number shows that there are 60 amino acids. This is just a continuation. Here, this is a continuation. And notice that we have some symbols. The asterisk represent that our amino acids that are identical in this particular position. So this asterisk means that the amino acids here are all identical. We have F in the whole column. For example, here in another asterisk, all column is N, and so on. Now, for easier comparison, let us push on the show color buttons. And notice now that the amino acids are color-coded. So what the colors represent are the amino acids that are similar. For example, now here, if they are in the same color, they are either identical or very similar. The column here means that there's a conservation. It indicates a conservation between groups of strongly similar properties. And the period indicates conservation between groups of weakly similar properties. And now if you click on this button, phylogenetic tree, you will construct the phylogenetic tree, which we will analyze in a moment. This is the phylogenetic tree of our species. Before we do the phylogenetic tree analysis, we will define something that is called molecular clock. Molecular clock is a figurative term for a technique that uses the mutation rate of biomolecules to deduce the time in prehistory when two or more life forms diverged. So simply, it is a tool used to calculate the timing of evolutionary events, and what does it really measure is the number of changes or mutations which accumulate in the gene sequences of different species over time. Now, going to the phylogenetic tree as a definition, it is a branching diagram or a tree showing the evolutionary relationships among various biological species or other entities. It is based upon similarities and differences in their physical or genetic characteristics. Now, why it is useful? Because it helps in knowing the evolutionary history of organisms, and it shows how and when the branches of the phylogenetic tree have evolved from the common ancestor. Now, let's know more about the anatomy of the phylogenetic tree. In a phylogenetic tree, the species or groups of interest are found at the tips of lines referred to as the tree's branches. For example, the phylogenetic tree below represents relationships between 5 species, A, B, C, D, and E, which are positioned at the ends of the branches. The pattern in which the branches connect represents our understanding of how the species in the tree evolved from a series of common ancestors. Each branch point, which also called an internal node, represents a divergence event or splitting apart of a single group into 2 descendant groups. At each branch point lies the most recent common ancestor of all the groups descended from the branch point. For instance, at the branch point giving rise to species A and B, we would find the most recent common ancestor of those 2 species, and at the branch point right above the root of the tree, we would find the most recent common ancestor of all the species in the tree, A, B, C, D, and E. Another important term to know in the phylogenetic tree is the clade, which is the branch that includes a single common ancestor and all of its descendants. The relatedness of 2 species has a very specific meaning in the phylogenetic tree. Two species are more related if they have a more recent common ancestor, and less related if they have less recent common ancestor. We can use a pretty straightforward method to find the most recent common ancestor of any pair or group of species. In this method, we start at the branch ends carrying the two species of interest and walk backwards in the tree until we find the point where species lines converge. For instance, suppose that we wanted to say whether A and B or B and C are more closely related. To do so, we would follow the lines of both pairs of species backward in the tree. Since A and B converge at a common ancestor first as we move backward, and B only converges with C after its junction point with A, we can say that A and B are more related than B and C. Going back to our example phylogenetic tree, here Saccharomyces and Arabidopsis are in one clade, and the internal node from which they diverge represents the most common recent ancestor of the two species. Also, Saccharomyces and Arabidopsis have the same ancestor with Drosophila. Now, because Saccharomyces and Arabidopsis have a more recent common ancestor, we can say that they are more related than Arabidopsis and Drosophila, since they have less recent common ancestor. This tree is rooted, and the root is the common ancestor of all species. Now, because it is rooted, this suggests that we know where the ultimate common ancestor of the all species was. Knowing this gives the tree an order of branching events in the horizontal dimension. So ancestor A exists prior to ancestor B and C, and time is approximately flowing from left to right. This brings me to the end of my presentation. We talked about multiple sequence alignment and its applications. We used also Clustal software to perform multiple sequence alignment, and we know a little bit about the phylogenetic tree. I hope you enjoyed the presentation and you get the benefit. Thank you.